cytoskeleton. The protein components of the cytoskeleton, chitos, cell, and skeleton dried body, interconnect and extend from the nucleus to the plasma membrane in eukaryotic cells. Prior to the 1970s, it was believed that the cytoplasm was an unorganized mixture of organic molecules. Then high-voltage electron microscopes, which can penetrate thicker specimens, showed instead that the cytoplasm was highly organized. The technique of immunofluorescence microscopy identified the makeup of the protein components within the cytoskeleton network. The cytoskeleton contains actin filaments, intermediate filaments, and microtubules, which maintain cell shape and allow the cell and its organelles to move. Therefore, the cytoskeleton is often compared to the bones and muscles of an animal. However, the cytoskeleton is dynamic, especially because its protein components can assemble and disassemble as appropriate. Apparently, a number of different mechanisms regulate this process, including protein kinesis that phosphorylate proteins. Phosphorylation leads to disassembly, and dephosphorylation causes assembly.
Actin filaments. Filaments, formerly called microfilaments, are long, extremely thin, flexible fibers about 7 nanometers in each diameter. That occur in bundles of mesh-like networks. Each actin filament contains 2 chains of globular actin monomers twisted about one another in a helical manner. Actin filaments play a structural role when they form a dense complex web under the plasma membrane to which they are anchored by special proteins. They are also seen in the microvilli that project from the intestinal cells. That presence most likely accounts for the ability to microvilli to alternately shorten and extend into the intestine. In plant cells, actin filaments apparently form the tracks along which the chloroplasts circulate in a particular direction. Doing so is called cytoplasmic streaming. Also, the presence of a network actin filaments lying beneath the plasma membrane accounts for the formation of pseudopods, extensions that allow certain cells to move from an emboid fashion. How are actin filaments involved in the movement of the cell and its organelles? They interact with the motor molecules, which are proteins that attach, detach, and reattach. Farther along the actin filament, in the presence of ATP, the motor molecule myosin pulls actin filaments along in this way. The myosin has both a head and a tail. In muscles, the tails of several muscle myosin molecules are joined to form a thick filament. In non-muscle cells, the cytoplasmatic myosin tails are bound to membranes, but the heads still interact with the actin.
The green portion is actin subunits. From the left, it begins with the tail, the head, and then the membrane.
Intermediate filaments. Intermediate filaments, 8 to 11 nanometers in diameter, are intermediate in size between actin filaments and microtubules. They are rope-like assembly of fibrous polypeptides. By the specific filament type varies according to the tissue. Some intermediate filaments support the nuclear envelope, whereas others support the plasma membrane and take part in the formation to cell-to-cell junctions. In the skin, intermediate filaments made the protein keratin give great mechanical strength to skin cells. We know the intermediate filaments are highly dynamic and will disassemble when phosphate is added to them by kinesis. Microtubules. Microtubules are the small hollow cylinders about 25 nanometers in diameter from 0.2 to 25 in length. The microtubules are in globular protein called tubulin, which is 2 types called alpha and beta. There is slightly different amino acid sequence in alpha tubulin compared to beta tubulin. When assembly occurs, alpha and beta tubulin molecules come together as dimers, and the dimers arrange themselves in rows. Microtubules have 13 rows of tubulin dimers surrounding what appears in electron micrographs to be empty central core. The regulation of microtubule assembly is under the control of microtubule organizing MTOC. The regulation of microtubule assembly is under the control of microtubule organizing center. In most eukaryotic cells, the main MTOC is the centrosome, which lies near the nucleus. Microtubulus radiates from the centrosome, helping to maintain the shape of the cell, acting as tracks along which organelles can move, whereas the motor molecule myosin is associated with actin filaments. The motor molecules kinesin and dynein are associated with microtubulus.
Figure 4.18, the cytoskeleton. The cytoskeleton maintains the shape of the cell and allows its parts to move. Three types of protein components make up the cytoskeleton. They can be detected in cells by using a special fluorescent technique that reveals only one type of component at a time. A to left, or left to right. Animal cells are treated so that actin filaments can be microscopically detected. The drawing shows that actin filaments are composed of a twisted double chain of actin subunits. The giant cells of the green Algochara rely on actin filaments to move organelles from one end of the cell to another. Animal cells are treated so that intermediate filaments can be microscopically detected. The drawing shows that fibrous proteins account for the rope-like structure of intermediate filaments. A peacock's colorful feathers are strengthened by the presence of intermediate filaments. C. Left to right. Animal cells are treated so that microtubulus can be microscopically detected. The drawing shows that microtubulus are hollow tubes composed of tubulin dimers. The skin cells of a trillion rely on microtubulus to move pigment granules around so that they take on the color of their environment.
Centrolas are short cylinders with a 9+0 pattern. 9+0 pattern of microtubule triplets. 9 sets of triplets are arranged in our outer ring, but the center of the central does not contain a microtubule. In an animal cells and most protists, centrosome are two centrals lying at a right angles to each other. A centrosome, or as mentioned previously, is a major microtube organizing center for the cell. Therefore, it is possible the centrals are also involved in the process by which microtubulus assemble and disassemble. Before an animal cell divides, the central replicate, and the member of each pair are at the right angles to one another. Then each pair becomes part of the separate centrosome. During the cell division, the centrosomes move apart and are most likely to function in organized mitotic spindle. In any case, each new cell has its own centrosome and pair of centrals. Plant and fungal cells have equivalent of a centrosome, but the structure does not contain centrals, suggesting that centrals are not necessary as assembly of cytoplasmic microtubules. A basal body is an organelle that lies at a base of cilia and flagella and may direct the organization of microtubules within these structures. In other words, a basal body may seem for a cilium or flagellum what the centrosome does for the cell. In the cells and the cilia and flagella, the centrals are believed to give a rise to basal bodies.
Cilia and flagella. Cilia. Cilium, eyelash, hair, and flagella. Flagello, whip, are hair-like projections that can move either in an undulating fashion like a whip or stiffly like an oar. If a cell is not attached, cilia or flagella move the cell through liquid. For example, unicellular paramecia are organisms that move by means of cilia, whereas sperm cells move by means of flagella. If the cell is attached, cilia or flagella are capable of moving liquid over the cell. The cells that line up upper respiratory tract are attached and have cilia that sweep debris trapped within the mucus back up into the throat, where it can be swallowed. This action helps keep the lungs clean. In eukaryotic cells, cilia are much shorter than flagella, but they have a similar construction. Both are membrane-bounded cylinders enclosing a matrix area. In the matrix are 9 microtubulate doublets, arranged in a circle around 2 central microtubulus. This is called 9+2 pattern of microtubulus. Cilia and flagella move when the microtubulate doublets slide past one another. As mentioned, each cilium and flagella have a basal body lying in the cytoplasm at its base. Basal bodies have the same circular arrangement of microtubulate triplets as centroles and are believed to be derived from them. It is possible that basal bodies organize the microtubulus within cilia and flagella, but this is not supported by the observation that cilia and flagella grow by the addition of tubulin dimers to their tips.
In a non-dividing animal cell, there is a single pair of centroles. In the centrosome located just outside the nucleus, just before the cell divides, the centroles replicate, producing 2 centrosomes. During cell division, the centrosomes separate so that each new cell has 1 centrosome containing 1 pair of centroles, as shown in the image. This is 1 centrosome with 1 pair of centroles. This is 2 centrosomes with 2 pairs of centroles. List the components of the cytoskeleton.
The cytoskeleton contains 3 important things: actin filaments, intermediate filaments, and microtubulus. The actin filaments, though the tiniest filament, interacts with the motor molecule myosin. It looks green with multiple subunits. Intermediate filaments support the nuclear envelope and the plasma membrane. It looks blue and ropey. Microtubulus are present in central.
Explain the structure of the cilia and the flagella. The cilia and flagella serve as a track for other molecules, and they are orange and in a spiral shape. Give an example of cell that has a cilia and one that has a flagella. Describe the functions of these cells. A eukaryotic cell contains the cilia. In the eukaryotic cell, it contains the nucleus, which contains the home for all genetic data.
Structure of flagellum. Below left. The basal body of the flagellum has a 9+0 pattern of microtubulate triplets. Notice the ring of 9 triplets, with no central microtubulus. Above left. In sperm, the shaft of the flagella has a 9+2 pattern. A ring of 9 microtubulate doublets surrounds a central pair of microtubulus. Middle right. In place of the triplets seen in a basal body, a flagellum's outer doublets have side arms of dynein, a motor molecule. Below right. In the presence of ATP, the dynein side arms reach out and attempt to move along their neighboring doublet. Because of the radial spokes connecting the doubles to the central microtubulus, bending occurs.
In summary, the cytoskeleton contains actin filaments, intermediate filaments, and microtubules. These maintain cell shape and allow it and the organelles to move. Actin filaments, the thinnest filaments, interact with the motor molecule myosin in muscle cells to bring about contraction. In other cells, they pinch off the daughter cells and have dynamic functions. Intermediate filaments support the nuclear envelope and plasma membrane and probably participate in cell-to-cell junctions. Microtubules radiate out from the centrosome and present in centroles, cilia, and flagella. They serve as tracks with which vesicles and other organelles move due to the action of specific motor molecules.